Hey Bio30s, I'm back again for another lesson. Today we're going to talk about abnormal meiosis and what occurs during nondisjunction. So, when we talk about nondisjunction, we're going to discuss what happens when something goes awry with meiosis. So for example, if two homologous chromosomes stick together and they don't split up during anaphase, either anaphase 1 or anaphase 2, you will end up with a situation where one daughter cell will have one extra chromosome and one daughter cell will be missing a chromosome. If you lack genetic information or have too much genetic information, this will cause your cells to not function properly. Remember, if you start off missing a chromosome or having too many chromosomes, in your zygote, when that zygote divides into 2, 4, 8, 16 cells and further on, every single cell in your body will have that abnormality. So every single cell will have 45 chromosomes, or every single cell will have 47 chromosomes. So let's talk about how it happens. So let's say for example we have anaphase 1 during meiosis 1. And let's say that right here, this homologous pair, that tetrad, doesn't split up. So it doesn't split up at all, it sticks together. That means that when we finish, this cell right here will contain an extra chromosome. And this cell over here will be missing a chromosome. Then we'll undergo meiosis 2, and for simplicity's sake, let's just say this is sperms. These are four sperm cells. This sperm cell would have an extra chromosome, so it would have 24 chromosomes. This one would also have 24, and then these two would have 22. So that's 4 abnormal sperm cells produced by this nondisjunction. So let's say, for example, one of these sperm cells were to win the great sperm race and go ahead and fertilize an egg. What would happen is, if one of these two sperm do it, that zygote would have 47 chromosomes. And if one of these sperm cells win, that zygote would have 45 chromosomes. Meaning, you're either lacking genetic information or you have too much genetic information. And depending on which chromosome it is, will then be related to what syndrome or symptoms you might experience because of this. So let's say, for example, this occurs during meiosis 2. So meiosis 1 goes without a hitch, right? Both of these cells are fine. But then during meiosis 2, let's say that chromosome doesn't split up. Remember, since we're talking meiosis 2, we're talking about chromatids splitting up, not chromosomes. That would result in this cell right here having an extra chromosome, so that'd be 24. This cell right here lacking a chromosome, so that'd be 22. But the two cells over here would be normal, each having 23 chromosomes. So you see that depending on what stage of meiosis nondisjunction occurs can impact how many gametes are abnormal because of it, right? So, if we talk about the two disorders I kind of mentioned already, the scientific names are trisomy or monosomy. So a trisomy disorder would be a disorder where you have an extra chromosome. So you have 24 chromosomes plus a regular 23, giving you 47 chromosomes, so one extra chromosome. If you have a monosomy disorder, this would be 22 plus the 23, giving you 45 chromosomes, you're missing one, okay? Now, this is what we call a karyotype. So a karyotype is essentially a snapshot or a picture of your chromosomes, and they're always arranged into homologous pairs. So I like to think of them as seats on a bus. So there's seat 1, seat 2, seat 3, seat 4, seat 5, and so on and so forth. So there's 23 seats on the bus that is your cells. And in each seat, you'll always have 2 chromosomes, one from mom, one from dad. So every seat here is full with 2 chromosomes. The last seat would be your sex chromosomes, either XY or XX. In this case, it's an XX, so this would be a female with a normal amount, 46 chromosomes. Now, how do you obtain a karyotype? Well, it's pretty straightforward. You'll take some type of culture of cells, whether that be blood cells, amniotic fluid, chorionic villi. You'll run it through a centrifuge. You isolate the cells. You want to isolate them during cell division so you can actually see the chromosomes condensed. Remember, during interphase, those chromosomes are uncondensed. Very, very difficult to see. So you're going to see the condensed chromosomes, and then what's going to happen is you're going to get a picture of these chromosomes, maybe add some stains so they're easier to see, and then we'll arrange them once again into their pairings. And you'll match them up based on shape, size, location, orientation, things like that. Now, a couple things to talk about here is, well, what happens if you do have non-disjunction in one part but not the other? So for example, what happens if you have an extra chromosome here or an extra chromosome there or missing one here? It gives you different syndromes or different disorders. So for example, Down syndrome is a trisomy condition. We refer to it as trisomy 21 because a person with Down syndrome has an extra 21st chromosome. So they would have trisomy 21. Usually someone with Down syndrome would have some type of mental or cognitive delays, but in terms of health, no real major health issues, I suppose. The risk of having a child with Down syndrome will increase significantly as a mother gets older, because what happens is as a mom gets older, maybe into her 40s, later 40s, things like that, her eggs are older as well. That means that there's a higher chance that a non-disjunction would occur, and we get an egg with an abnormal number of chromosomes. Before I move on there, here's a karyotype for someone with Down syndrome. You'll notice that all bus seats are full, except for the 21st seat, there's an extra spot, right? There's 1, 2, 3 chromosomes in that bus seat. So it's a trisomy condition with 3 chromosomes in that bus seat. Next disorder I want to talk about here is Turner syndrome. So Turner syndrome would be a monosomy disorder where you have a single X chromosome. So an individual with a single X chromosome would be fertilized, say, by a normal sperm, and then the egg wouldn't have any X chromosomes. So that egg would be blank in terms of sex chromosomes. The sperm would come in, deliver an X chromosome, and this person would have 45 chromosomes. They would only have that single X. Now, usually this occurs in 1 in every 10,000 births. When they do get born, most don't develop sexually because you'll need the 2 X chromosomes to actually undergo that secondary sex characteristic change during puberty. A question I always get is, okay, if there's a single X, is it possible to have a single Y? And the answer is no. You need at least 1 chromosome to have a viable zygote, right? The X chromosome contains a lot more information than just sexual, reproductive organs, things like that. The X chromosome does contain some very important information, whereas the Y chromosome doesn't contain very much information. It's very small, and on its own, it would not be able to support a full-blown zygote, let alone an embryo or a fetus. So, yes, it is possible to have 1 X chromosome, but it's not possible to have 1 Y chromosome. Then, of course, you can have XX, you can have XY, and moving forward into, say, trisomy, you could have someone who's XXY. So, for example, maybe we have a non-disjunction in a sperm cell where we have an X and a Y together, and then it fertilizes an egg with an X chromosome. We now have an individual with 2 X chromosomes as well as a Y chromosome. This would be a trisomy condition called Klinefelter syndrome. So these individuals would appear male at birth, but then later develop female sex characteristics due to high levels of estrogen during puberty. Usually occurs in 1 in every 1,000 births, and once again you would inherit 2 X chromosomes as well as a Y. There is a situation also where you could inherit 3 X chromosomes. This is called triple X syndrome. So you would have, say, 2 X's in the egg, 1 X from the sperm, giving you 3 X's. No real issues there. There just might be a little more in terms of hormones and things like that. It may lead to sterility and being unable to have kids, but in terms of health effects, no major concerns there. 2 other disorders I want to talk about before we end. There's Edwards syndrome and Pitot syndrome. Both are pretty significant disorders, Edward being a trisomy 18, so you have 3 chromosome 18, and Patau's syndrome being a trisomy of the 13th chromosome. Both of these involve major health defects. So heart problems, organ problems. Usually babies are born smaller heads, smaller jaws, not developed fully. And a lot of problems related to their health. Most babies, unfortunately, with Edward or Patau syndrome, if they are born at all, don't live very long, maybe not even a year. Some do live longer, but for the most part, it's a rare case that these babies would live very long. So what I want to get at here to end is you'll notice that things like trisomy of the 18 or trisomy of the 13, those chromosomes that are not sex chromosomes, have a way larger impact on your health than, say, a sex chromosome trait would, right? Because if you're Klinefelter or Turner syndrome or triple X, in terms of health, you're not doing too bad. But in terms of Edwards or Patau syndrome or other syndromes out there, if you have a monosomy or trisomy with those chromosomes that are 1 to 22, they tend to have a lot more health problems than the other ones. So that's the end of chapter 17. We're going to jump ahead next lesson to chapter 20, talk about DNA and how DNA replicates itself. So thanks for watching, and we'll chat soon.